Hello guys and welcome back to Exocytosis Academy. So today we are going to do the last topic of biological membranes. Okay. So before I proceed, I just thought of, just checking the concepts clarity of you guys. So one question I have got over here. So glucose is frequently administered intravenously to patients as a food source. So I hope you know what is intravenously, like directly into the blood, IV. Okay. Given that the transformation of glucose to glucose 6-phosphate consumes ATP, why not administer glucose 6-phosphate directly instead? Yes.
Ma'am, because glucose 6-phosphate can't get inside the cells.
Why it can't get inside the cell?
Because the action of converting glucose to glucose 6-phosphate is supposed to hold the glucose back from exiting the cell.
Yes, very good. And we can add more to this. So this, what you have said is the answer of this part. Like I'll tell you why. So Shruti is saying that glucose is outside, right? It comes inside to form and then it becomes glucose 6-phosphate. Now what she said is that it becomes glucose 6-phosphate and then it cannot go back, right? That's what Shruti you said?
Yes, ma'am.
Yes. Okay. Now the question is asking, it is correct. What you said is absolutely correct. But again, if I'm saying that I am giving to the patient directly glucose 6-phosphate, right? Why can't the patient can consume this? ये तो सही है कि ग्लूकोज हमने दिया, ग्लूकोज 6-फॉस्फेट बना ताकि वो वापस बाहर ना जाए. ओके, बट अगर मैं ग्लूकोज 6-फॉस्फेट ही डायरेक्टली दे रही हूँ तो क्यों कंज्यूम नहीं कर पाता, ये सवाल है.
Because the same thing applies, right? So if glucose 6-phosphate can't get outside, it also can't get inside.
Okay, now why it can't get outside? Same way. Shruti, if you know the answer. Okay.
Yeah.
Okay.
The phosphate group makes the difference over there. That is what causes the membrane to not be able to, as in glucose to not be able to pass through the
Very good.
glucose
Very
transporter.
good. But again, what does phosphate is doing to glucose and making it unable to cross the membrane? चाहे इस side हो या इस side हो.
The transporter is only accepting a certain type of chemical. The phosphate group is changing its chemistry as well as its stoichiometry so that it doesn't pass through the receptors.
Wow, that's good. Again, let's become more precise. Okay, so it's all correct what you said. Okay, so the transporter is only available for our glucose molecule. What we have discussed before, GLUT transporters 1 to 12 are there. It is only meant for your glucose transporter. Okay, sorry, glucose transport. Now, if it becomes glucose 6-phosphate, then it does not have affinity for glucose 6-phosphate. Absolutely correct. Now, there's one more thing which phosphate is doing to the glucose. What is it?
Adding charge.
Correct. And making it?
Bigger.
Correct. And there's one more word. So, with respect to size, as we know that through the membrane, okay, when we talk about semi-permeable membrane, the permeability of glucose, first, it has got increase in the size. That's why it cannot go. Now, the addition of phosphate is also giving the charge. So, increasing the charge in the glucose, it's also increasing its dash. Glucose is what kind of molecule?
Sugar.
Sorry, nahi baba. It is hydrophobic, hydrophilic.
Hydrophilic.
Correct. So when you add anything charge, चाहे negative डालो या positive डालो, what is happening? It is increasing its hydrophilicity. Correct? It is making it more polar. Now crossing of this highly bigger in size plus highly polar molecule across the membrane is not possible. And that's why at the hospitals we give glucose and not glucose 6-phosphate. Okay? Now, along with the glucose, if you remember, in the saline, we say saline, और जब हम बोलते हैं ग्लूकोज चढ़ रहा है, ठीक है, तो ग्लूकोज के साथ हमेशा आपका Na+Cl- भी रहेगा. If you remember, we have talked about this importers. ग्लूकोज के साथ Na+ भी चाहिए, and that's why saline चढ़ाते हैं. क्यों? Because it gives easy absorption of glucose in the intestinal lumen. If you remember, that figure was there where glucose is getting accepted by the glut transporters, but it should reach, like it should get absorbed in the intestine. We need Na+ also. तो जो हमारा Na+ glucose का importer था, it is helping. Now this is the reason why in whenever you feel dehydrated या चक्कर आता है, हमेशा आपको नींबू, नमक, चीनी का मिक्स देते हैं. ORS क्या है? It is just a mixture of salt, sugar. Correct? So this is the reason that only glucose is not given. Along with that, Na+ is required. Okay, so that was really good. It was beautiful having this discussion. Okay, good. You won't believe, guys, I am feeling proud with this discussion. Like, it was such a nice way of explaining. Good. Thank you, Shruti. Okay, I want Akshay and Tanvi also to participate more. Okay, so only one question was there. Let's talk about the transport and transfer across the cell membranes in bulk. So this is the last topic here. Till now, we have discussed that in the with respect to the channels, correct? Wherein which we were talking about single individual ions or single molecule like sugar हो गया आपका, जैसे glucose का हो गया, right? Now what we are going to discuss is like how the entire in bulk or cargoes are actually getting transported across a membrane. There are two ways by which exocytosis and endocytosis. So this is something which already you guys know. Exo क्या है? Exo is something which is going out of the cell, correct? If you remember in the ligand gated channel, example of acetylcholine receptor, we have talked about this, right? That how acetylcholine neurotransmitters are getting released out in the synaptic cleft, so that from the presynaptic neuron, it can bind to the postsynaptic neuron. If you remember, guys, यहाँ से आता है, release होता है, and here you have got your acetylcholine, right? So this go and binds to the receptors present over here. Now this exocytosis and endocytosis, now we are going to discuss. Very simple, very small topic. Okay, so exocytosis क्या हुआ? The fusion of vesicles or vacuoles with the plasma membrane, resulting in the secretion outside the cell or discarding waste outside the cell. ठीक है? So for example, if you talk about the growth hormones also, okay, or let us talk about insulin. So even insulin is getting released via the process of secretory vesicle. Okay, and they go outside. And when this insulin reaches the target area, it gives a signal of more expression of glut transporters so that the glucose can get absorbed by the cell. ठीक है? कहाँ से? From the blood, so that your blood does not have the glucose present. Now, what will happen if you have got glucose present in blood for longer duration? क्या होगा? Of course, this happens in the case of diabetes. But what damage does glucose is doing in the blood?
It binds to hemoglobin.
No. और सोचो. और सोचो, it is not the hemoglobin protein, but something else. It does bind to the other proteins. Can you guess which is the most abundant protein in the blood apart from hemoglobin? Hemoglobin से भी ज्यादा ही है वो actually. No idea. Tanvi, would you like to try? Okay, no issue. See, have you heard about albumin?
Yes.
Yes. So it is present in our blood serum, right? Now, what happens over
Yeah.
here is that there is something called, this is just kind of extra information, not extra, but it is important. तब क्या होगा हम ये फिर physiology में cover नहीं करेंगे because it's very easy to understand also. There are two types of chemical reactions, glycosylation and there's something called glycation. ठीक है? Now, what is glycosylation? Have you heard of it? आपने सुना होगा glycosylation. There are two types are there, O-linked and N-linked. ठीक है? O-linked means the sugar moieties are attached to the serine or threonine because they have got the OH group. Okay. N-linked means it is asparagine wherever you have got the N. So उसको अगर आप attach करते हो then it is called N-linked or O-linked respectively glycosylation. So this kind of glycosylation is like programmed. It means what? It is followed by the enzymatic steps, like proper steps के साथ आपका addition of glucose is taking place. Okay. Whereas glycosylation, okay, glycosylation, sorry, not glycation, that is glycosylation. Now, glycation is what? Glycation is something which happens randomly. Okay. And it is not either O-linked or N-linked. This is the binding of glucose forming a covalent bond randomly on a protein. Now, what will happen if randomly anywhere the proteins are getting bound with glucose? This will result in the misfolding of the proteins. Correct, because आपने कहीं पे भी bind कर दिया, it is going to change the structure of the protein. Now, if there is a structural change, what will happen to the function of the protein? It will lose its function. Correct? Agree? Now, because of this, what happens? जब ग्लूकोज आपका ब्लड में बहुत देर से प्रेजेंट है, okay, and if it is not taken up by the cells or the tissues, then this glucose gets randomly glycated on the albumin proteins and the other proteins. Okay. हीमोग्लोबिन क्या है? हीमोग्लोबिन इज लाइक इनसाइड द आरबीसीज एंड ऑल, राइट? तो इतना इफेक्ट नहीं करेगा. बट if it is present outside in the blood, okay, then what happens? Whatever the albumin proteins or other serum proteins are there, it's not only albumin, many other proteins are there. We have got this platelet deriving factors are also there, right? If all these proteins are getting glycated to the glucose, so all of them are undergoing misfolding, lose of the function, right? Eventually what is happening? And that's why whenever any wound is there, there is no healing. And you know, in the case of diabetes, it is true, right? There will be a delay or no healing of the wound. So this is why the glucose is affecting the proteins in the diabetic patients. Okay? Okay, we just spent little away from the topic. So coming back over here, so I was explaining about the insulin release, correct? So ये कैसे होता है? You get the gene expression, right? mRNA is synthesized from the mRNA. It goes for the translation of insulin. So insulin is your polypeptide hormone. Now, insulin, it is there in the ER now. So insulin has got the signal peptide, which is giving the information to the cell that it should be released outside. That is, it should be extracellular secretion. Okay. So the ER is going to process a signal peptide. It gets properly folded in the ER and in the Golgi body. And then that signal, okay, it is detected by the secretory vesicles. They understand and they take this vesicle to the plasma membrane where it gets fuses and releases the insulin. Okay, so this is one example. The other example we have seen in the case of the acetylcholine neurotransmitters. Guys, are you able to follow this very simple topic? As such, कुछ याद करना भी नहीं है इसमें. This is the same thing. So you have got here ER and Golgis are there, right? The proteins are there. Now these proteins, are present in the transport vesicle, they go and directly go and fuses with the plasma membrane. Okay. Then again, two types are there. You have got the constitutive secretion and you have got the regulated secretion. Constitutive kya hoga? That every time it is produced and released. Every time it is produced and released. Whereas regulated means only when the signal is given. Okay. Gene expression hoga, tabhi wo synthesize hoga. Otherwise, no. Okay. Now, having understood the concept of the constitutive secretion and regulated secretion, now tell me, insulin comes under what category? Is it constitutive or regulated?
Regulated.
Regulated.
Very good. It is regulated. Why? Because whenever this information is given to the cell that we need to absorb glucose right now, and for which insulin is required, then only the gene expression is happening, right? Good. Now, example of constitutive. So constitutive expression means or secretion means every time it is required. तो ऐसा कौन सा एंजाइम या प्रोटीन है जो हमेशा हमें रिक्वायर्ड रहेगा?
Acetylcholine.
Acetylcholine is not a protein, right?
Neurotransmitter.
हाँ.
Albumin.
Yes, albumin is also there. Correct. Many are there. ऐसे तो बहुत है. For example, there are many proteins like so we are talking about the secretion. Hello. Yes.
Saliva secretion.
Yes. जो भी enzymes हो गए आपके amylase, right? अभी जब तक सेल के अंदर रहेंगे, it is not going to help us. So all these enzymes which is required for the digestions are all for the constitutive secretions. ठीक है? Okay, good. Now, okay, it's the same thing what we have discussed. So molecules like neurotransmitters and proteins are transported by secreting them through an energy-dependent process. Yes, Shruti, this is also correct. Now, in exocytosis, membrane-bound secretory vesicles are carried to the cell membrane and the contents are secreted into the extracellular environment. Now, the secretion is possible because the vesicle fuses with the plasma membrane. Okay. Now, it is also a mechanism by which cells are able to insert membrane proteins. Yes, this also I wanted to explain. I do. Okay, I don't think I have a figure. So, see, let us say that your proteins are synthesized. Let's say that we are talking about the GLUT transporters. Okay, so GLUT transporters are nothing but again the proteins, correct? So, but they are your membrane bound proteins. So, when these are synthesized, okay, in the ER endoplasmic reticulum bound ribosomes, ठीक है? अब उसमें क्या होगा? These ribosomes, these GLUT polypeptides will be produced and they will be secreted into the lumen of the ER. Now, this proteins forms a vesicle, goes to the Golgi, where it gets modified. But always remember that these vesicles, यहाँ पे ही आपका GLUT transporters present है. They are never in the soluble form. Membrane bound रहेंगे and यहीं पे ही आपका structural folding होगा. Okay. Now from here, from the Golgi, it is in the vesicles only, right? Secretory vesicles में रहेंगे. It's like this. Your GLUT transporters will be like this. Now these vesicles will go. Let us say that this is your plasma membrane. Now here they get fused. So what will happen when they get fused? At the end, you're the membrane. Membrane का जो length होगा वो आपका increase हो जाएगा because this much secretory vesicle has been added over here, right? And that's how the polypeptide chain or the GLUT transporters are exposed on the plasma membrane. Okay? So this is how it is helping in inserting the membrane proteins. Did you get this point? Now, here I can ask one question. इतना fusion होता है of secretory vesicles of the plasma membrane. still we can see that the size of the cell remains the same. How? Even though these many fusions are taking place, अब देखो आपने इतना ऐड किया है तो इतना आपका लेंथ बढ़ गया प्लाज्मा मेम्ब्रेन का, but when you check it is always the same. मतलब इतना सरफेस एरिया होना चाहिए तो इतना ही रहता है हमेशा. Why and how it is maintaining? Yes.
Ma'am, maybe the rate of endo as well as exocytosis is both similar, so they're losing just as much as they're gaining.
Yes, yes, correct. So even if it is not taking anything, like endocytosis क्या होता है? Where you engulf something or you take from the outside. In order to maintain the same surface area to volume ratio, the vesicles comes out. They pinches off inside. ठीक है? And then same vesicles go and fuses with the ER and Golgi. तो इसलिए हमेशा from ER to Golgi, Golgi to ER, Golgi to plasma membrane to Golgi, it all always happening, right? Because of this, they are able to maintain the constant ratio. Okay, good. Now, types of exocytosis, we have got two types based on the calcium. So we have got the calcium triggered non-constitutive and non-calcium triggered constitutive. ठीक है, in short regulated and non-regulated. So calcium triggered क्या हो गया आपका, जो हम neurotransmitters की बात करते हैं. So it is the triggered non-constitutive exocytosis. It requires external signal in specific sorting signal on the vesicles like clathrin coat, as well as increase in the intracellular calcium. Now this is happening in the case of the neuronal chemical synapse. Now coming to the non-regulated, it is performed by all the cells which serves the release of components of the extracellular matrix or delivery of any membrane proteins. तो अभी हमने दो example देखे थे, proteins and neurotransmitters का. So neurotransmitters are the regulated one and the proteins ones are the non-regulated one. Okay. Now look at the figure here. One second. Okay, so let's talk about the first, the constitutive ones. So what is happening in the constitutive one first? Let us say that the neurotransmitters are getting released. Okay, now if you remember in the previous figure also where we were talking about the ligand receptor, the acetylcholine neurotransmitter, there also we saw that in order to carry out this release of the neurotransmitter in the synaptic cleft, calcium uptake was there via the calcium channels. Okay, excuse me. Now, so when these neurotransmitters are synthesized, they form the vesicles. Okay, now they keep on increasing the calcium level. Now once it is done, then they get fused with the plasma membrane and released into the exocytosis. Okay, in the regulated secretion where the calcium is required, and in the protein that is directly. Now this expression, this secretion, it can be endocrine, paracrine, and autocrine. So endocrine means, for example, it is in the case of the hormones. Okay. Sorry. Okay, autocrine is for the self signaling, जो भी हमने कल देखा था in the case of the toxin which we were studying about, right? Interleukin 12, calcimycin effect, right? The paracrine is nearby cell, the signaling. Oh God, yeah, so sorry. Yeah, now coming to the endocytosis. Now this endocytosis is your energy dependent process and lot of amount of ATP is required. Yeah, so here what we need to remember is, this is really important guys, okay, as compared to the exocytosis, endocytosis is more important because you need to remember all these proteins. Okay, now this is the inside of the cell, okay, and this is outside. Now what is happening here? So we see that there are the ligand molecules or the cargo molecules which goes and binds to the receptor. Now this binding of the receptor to its ligand is going to recruit the proteins called adaptin. Now adaptin क्या कर रहे हैं? Can you see it binds to the receptor? It is a connection between the receptor as well as the clathrin code. Clathrin are the trimer proteins which is responsible for pinching of the plasma membrane okay, and taking inside. Okay, now, so adapted proteins comes and binds to the receptor. To this adaptin, the clathrin code comes. So you can see it over here. Slowly, these clathrin code undergo structural change. ये कुछ ऐसे हैं, something like this. So when they bind to the adaptin, they start what to say? It's a contract करना शुरू कर देते हैं, जिसकी वजह से क्या होगा? They start forming a bulging kind of thing, right? Now, when they come together, the dynamin protein comes, and when with the help of the ATP, it twists. तो अभी क्या हो रहा है? कोई भी चीज को आप ज्यादा twist करते हो तो वो pinch off हो जाता है वहां से, correct? So dynamin protein is doing that, the pinching off of the plasma membrane. Then the clathrin molecule will come. Now this is your endocytose molecule, which will go inside of the cytoplasm, right? Now the clathrin protein will leave and only the vesicle is left. This vesicle required, अब इसने depending on the cargo what it is, it will go and fuse with the lysosome. Is it clear, guys? अब जितने भी हमने endosomes, lysosomes, autophagosomes जो हमने पढ़े थे in the previous lectures, just add the additions of these adaptin and clathrin code. So जब भी endocytosis हो रहा है, these proteins are coming into the picture, and before fusion to the lysosome, clathrin vesicles or the clathrin code, they are leaving the vesicle. Okay, because this vesicle membrane, plasma membrane, and the membranes of the lysosome, ER, Golgi is similar. The composition is same. तो उसमें वो fuse हो जाएंगे. ठीक है? This is how the whatever cargo they have taken, whether it is a protein or ligand or whatever it is, it goes and binds to the lysosome then. Now, are you able to link everything, guys? Are you able to follow? Yeah? Yes, Akshaya?
Yes, ma'am.
Okay, okay. Okay. So this is how endocytosis is taking place, okay? But whereas in the case of the, what to say, in the case of the exocytosis, there are other different types of proteins involved, okay? And it is little a complex mechanism. So I thought of taking that with the other topic, which will come when we will talk about the cell cytoskeleton. ठीक है, वहां पे मैं वो cover करूंगी because it is a very complex mechanism by which exocytosis is taking place. Because see, there the fusion of the membrane is happening, right? जैसा अगर ये है और एक vesicle है, so this has to fuse with this so that the inside content can be released outside, right? So that fusion mechanism involves a lot of proteins. So it will take time, and it will be in continuation with the cell cytoskeleton topic. So that's why I always take exocytosis there really. I can just give you a hint that it involves V snare, T snare, these kinds of proteins are there. Then snare itself has got, it's such a big name. It's the acronym of a protein, okay? So all these are there. So T means target, V means vesicle. तो ये आपका V snare protein यहाँ रहेगा, which goes and fuses with the T snare protein. Now उन दोनों को fuse करने के लिए भी you have got the different set of proteins. Okay, so we'll cover over there when we'll talk about the cytoskeleton, cell cytoskeleton, wherein which we'll talk about the actin, microtubules, intermediate filaments, etc. Okay. Now, endocytosis. So this is, one second. Oh God. So they are the receptor proteins in the plasma membrane, which is bind to the specific molecules causing protein conformational change. So, sorry. So when I say receptor mediated means where I have talked about the clathrin codes. Okay. वहां पे उतना ही आपको याद रखना है. Now, phagocytosis आपने सुना ही होगा, right? So we used to take the entire food particle or the bacteria engulfed totally. So this bacteria, let us say that this is a bacteria. So it will have its own certain receptors which will go and bind to the, it will have certain its own sugar moieties which will act as antigens or ligands will bind to the receptors present over here. And it will be engulfed inside. Okay? Phagocytosis is जहाँ पे large solid particles we are taking. Now, when we talk about pinocytosis, it is your liquid molecules. So हम इसको क्या बोलते हैं कि हमारा सेल भी पानी पीता है. That is like, taking a bulk amount of liquid molecules inside. and then again it fuses with the lysosome where it is being required. So this is just your transmission electron micrograph where you can see so many bulging is happening, right? It means the cell is taking so much amount of the materials which is dissolved in the extracellular matrix and taking inside. Okay. Nothing much in Pino and phagocytosis. Yeah. So thank you guys, and very happy to say that this first our unit is completed now. Any doubts you guys want to ask in the membrane transport? Like around 15 lectures we completed in our membrane transport. Anywhere, any doubts are there which you would like to ask? Yes, Shruti?
So far, no, ma'am.
Okay, good. What about Akshay and Tanvi?
No, ma'am.
Okay, I'll be stopping here now then.